I'll start with the talk proper. So I can start by introducing this book, which I've based a lot of. If you can't see it at the back, it's Cell Biology by the Numbers. And if you want a sort of more in-depth understanding of biology, then there's the Gene series of books. Now into the 12th edition. So the first one I read of these things was edition number 4, and that was already sort of about 2 inches thick. And these things are absolutely packed full of information. They do regularly take out stuff which is out of date. This book, this Cell Biology by the Numbers book, is a very interesting book if you want to understand not just what things are, but how many things there are. So there's a kind of a lot of really interesting little facts in this book. So I'll kind of hopefully regurgitate some of these little factlets as we go along, because some of them are quite interesting. Just to kind of understand what makes us and what makes the things we eat or whatever. It's all kind of part of the biology world. If you're in the Oxford area, we have a Oxford Biohackers meetup, and of course the Oxford ACCU meetup, which we go to fairly regularly. I think there's going to be a talk in Oxford after as a deconstruction of this conference. So feel free to come along to those talks. So I think our next Biohackers is in the 6th of June, I think, and usually in the centre of Oxford. So, oops, I think I've lost my mouse pointer. Ah, mouse pointer. So we have, right, so a little comparison. So that is about the smallest transistor we can make at the moment. So that's, the dimensions of this is about 6 nanometers. In the space of a lithographic transistor, then we can get an entire CPU in the biological world. This thing is a ribosome, and that essentially is a CPU. It's quite a slow CPU, but we've got a lot of them. It acts a bit like a tape reader, and it reads some mRNA and squirts proteins out the top, and the proteins do most of the thinking. I'll kind of come to these in a second. So don't think of proteins as being your lunch. Proteins are complex little molecular machines. We have little pumps, we have little motors, we have things which can run along, towing things behind them, which you've probably all seen in videos. I'll show you that video in a second. And there's a whole bunch of, there's a lot of logic gates implemented in proteins. At all levels of the system. Probably about half of the proteins in your body, about 22,000 proteins in your genome, and about half of those are switching. They're implementing logic of various kinds. So, data storage. We'll start with DNA. So who discovered DNA? The structure of DNA. Franklin, there we go. Rosalind Franklin. And unfortunately, she didn't get the Nobel Prize because she was dead of all the radiation she suffered while she was discovering the structure of DNA. But her famous photograph 51 was seen by Crick and Watson, who then took all the credit. Admittedly, they did make a nice ball and stick model, which she didn't. So the amount of data in each of your cells is about 700 megabytes, which is about 3 billion base pairs. So A, C, T, and G. So each of these little rungs in the ladder is a base pair, and because there's 4 combinations of those, we get 2 bits per base pair. And that's about the equivalent of an old CD-ROM. In fact, when they first sequenced the human genome, I bought a copy of Prospect magazine. On the front was a CD full of the data from the human genome, and that was done largely at the Sanger Institute, amongst other places in Cambridge. So DNA is kind of read-only memory in a way, so it's like a ROM. So you can write it, but it's quite slow to write. Writing new data, copying it is really quick, but writing new data to DNA takes quite a long time. We can do that. We can make any DNA we like just by adding a sequence of chemicals in series, and you can kind of build any DNA you like. So you could use it as a very, very slow data storage medium, or as like a CD-ROM where you write your data once, then you can copy it as many times as you like. Copying DNA is extremely cheap, thanks to biology itself. So the diameter of that is about 2 nanometers, which is like sort of a fraction of the size of a transistor. And we get sort of about 5 bases per nanometer. So the density of the DNA is very high. So I think we have sort of, I can't remember, it's like this much DNA in each of our cells. And if you stretch it out end to end, then that's a strand which is 2 nanometers thick. And it holds 3 billion pieces of information. So it's doing quite well. Flash memory these days is doing fairly well. This, in my opinion, is doing a lot better because you can copy this massively in parallel. You can operate on it. You can build structures with it, and all this kind of stuff, which you can't do with flash memory. Flash memory is a bit brittle. If you twist it like this, it just breaks in half. So, but DNA, you can twist it and bend it and fold it and make all kinds of things out of DNA, not just store data, not just store data with it. I'll come to RNA in a bit, which is a bit more flexible. So we can, so viruses, for instance, HIV virus contains a bit of DNA. That's what it does. HIV virus is just a thing that takes a bit of DNA from one nucleus to another nucleus and transports it about. And DNA, what we most understand about DNA is the fact it encodes recipes for proteins. And proteins are like the one on the previous slide. There's lots of different types of proteins. And all those proteins do different things. They're not sort of blobs of stuff. Some of them are blobs of stuff that do chemical things, but a lot of them are very complicated shapes. And you've got motors and pumps and logic gates and all kinds of stuff we can make out of protein. So, big question is, how much data can we get in a pint of beer? And I'm talking real ale, not fizzy lager. So who likes their real ale in the room? So there you go. So we've got a few lovers of real ale. So who likes fizzy lager? You can find it out amongst yourselves later on. But, anyway, so would anyone like to guess how much data we can get in a pint of real ale? I'm saying real ale because the cells are actually, some of the cells are actually still alive. Very few, remarkably, because they get killed off with the alcohol. But in the remaining few cells, like the scum at the bottom of the beer, if you leave it to stand, how much data? Terabyte? Oh. Anyway, we're kind of do the math. So I'm kind of basing it on numbers in this book, which may or may not be correct. These are kind of back of the envelope calculations. So let's assume we've got 40 billion yeast cells. And I can't pronounce that. That's Saccharomyces cerevisiae, which is, and if you speak Spanish, you'll recognize the second word. That means beer in Spanish, and almost. And so it actually means beer in Latin. And this is the budding yeast. And probably without budding yeast, we wouldn't have lifted ourselves out of the Stone Age, because budding yeast forms a big part of our diet, and bread and beer and all these things we associate with coming of civilization came from this particular thing. It's a very well studied beastie is yeast, and it's got a medium. It's actually got a genome which is almost as big as ours. There are bigger genomes than ours. Like wheat, for example, has about 16 billion, has about 5 times bigger genome than we do. Partly because wheat defends itself with chemicals, and we defend ourselves. We have an immune system which not only keeps us alive, but it tends to trim off extra bits of. It tends to keep the code size of our genome relatively limited. So if new things pop up, they tend to get trimmed off by the immune system. Organisms which make new genes tend not to have a good time of it. So our immune system kind of trims out some of the extra genes we might make if we're now, of course, that's a sensible thing to do because viruses like to insert new genes in our genome. So being able to defend against that is kind of an important thing. So say we have 40 billion yeast cells, and say each cell it's about 12 million bases in a yeast cell, which is about 3 megabytes, which is smaller than our 700 megabytes, but it's still quite a bit of data. So now we're starting to kind of get a reasonable idea. We've got like 12 million bases, or 3 megabytes, and we've got 40 billion yeast cells. And 40 billion times 3 megabytes is 120 petabytes. And that's just the scum in the bottom of your beer. So imagine how much data there is in you. And so an average human has about 30 trillion cells. And we have about 700 megabytes per cell. So we're into exabytes when it comes to the human genome. The human genome has, but of course, the entropy of that data is very low. So like web logs, where the entropy is almost zero. That's why we need Hadoop, after all. Like web logs, most of that information just repeats. And so the cells in our body, actually the big variety of DNA in our body is our bacterial friends who play a big role in keeping us alive. You think of bacteria as being deadly, but actually your bacteria are really important in keeping you alive. To the extent that if you get antibiotics, so if you take antibiotics, which is actually a very dangerous thing to do, because it can breed resistant bacteria. So we've got Clostridium difficile, for example. If you do that, particularly if you're older, then you end up with only resistant bacteria in your gut. And so it's a complete monoculture of resistant bacteria. And that'll kill you pretty soon. Unless what you do is you take a bit of poo from a healthy person, and you take that poo and you insert it in the gut. I'm not going to tell you how to do that. You insert it in the gut of a person with C. difficile, and about two applications of that, you can cure almost the entire population of resistant bacteria. So it's kind of a miracle cure. It's just poo. Poo is the answer to everything, after all. That was the subject of the talk my wife did at the last Biohackers. Anyway, so moving on from poo. So the viruses and bacteria in your gut represent almost all the variety of information we have. So these days, if you really want to know what's wrong with someone, you take a sample of poo and you sequence it, and you find out what bacteria and what viruses are in your poo, and that will tell you an awful lot about. We typically have about 2 kilograms of bacteria we carry around in our gut. So it's a significant portion of our weight, and plays a very important role. So if you had to store 100, has anyone got 128 petabytes in their data set? Unless you're Google, you probably don't. We need 120 of these things full of 10 terabyte hard drives to store that. So that's quite a bit of data, and that's just your pint of beer. So nature has a sort of much bigger data managing problem than we do typically, but handles it very well. It's had a bit of time to do it. It's been around for a while. So next, I want to kind of do what is the RAM? What is the RAM? We've done what's the ROM of the biological computer. What's the RAM? So if you haven't been doing biology for the last 20 years or so, I think I studied biology at school about 30 years ago, just to date me. And it was all about dissecting rats and eyes and things like this. Not terribly exciting. And this kind of stuff, although people knew about it, it was difficult stuff, and we wouldn't teach kids this stuff. It's too difficult. But this is actually how all of life works. So we start off with DNA, which contains the program for our biological computer. There's a little enzyme called RNA polymerase that zips along the DNA at quite an impressive rate and copies the DNA to a strand of RNA, mRNA, and this messenger RNA. RNA is like the DNA, but it's much more flexible, and it lasts only a very short period of time. So if you there's a lot of stuff around which just break up RNA fairly quickly. So typically these are very temporary. They're like RAM. You're kind of storing stuff temporarily, but then you're going to throw it away a bit later. Some of the RNA hangs around a bit longer. So some of the really old bits of your biology are made of RNA. So before the proteins, we hypothesized there was RNA, which did everything. And so if you just leave a piece of RNA around in an amino acid soup, it'll make another piece of RNA exactly the same as the previous one, or the opposite of the previous one. And so RNA is capable of replicating itself given the right environments. So that's the hypothesis of how life started in the first place. As long as we had an amino acid soup, which is literally what soup mostly is. If you have a beef stock, it's essentially amino acid soup. Then, if you kind of leave your RNA sitting around in beef stock, then and it doesn't get gobbled up by something else, which of course these days it will, then it'll just replicate itself. So once you've got a pattern of RNA, then it'll pretty much copy itself. And if it can fold itself up into different shapes, then it can do lots of different things. So. In particular, we got the thing on the first slide, which is the ribosome. The ribosome is like a little assembly factory for proteins. And what happens is these little funny-shaped lander things, they kind of match up with three of the bases of the DNA, and they're carrying around one amino acid on the top of them. And the ribosome kind of gets everything in the right shape, squeezes these two amino acids together, and then squeezes the protein out the top like a tube of toothpaste, and sometimes it just squeezes it out into the cytosol. Sometimes it squeezes it through a hole, and you get different effects. And then there's a whole bunch of proteins which kind of shape the protein, called chaperones, which chaperonins, I think, which kind of shape the protein in various ways and fold them up into a variety of different shapes. With simple life forms like simple bacteria, we don't have that. They're very simple. They just have very simple mechanisms, very small genome, which just do the basic protein building stuff. But in humans, there's a lot of complicated mechanisms for making customized components in our bodies. So, for example, genes don't just come as a single strip of RNA. They come as a kit of protein-making parts. So you get sort of this bit, which is the helix, and this bit, which is the active site, and this bit, which is and sometimes it can construct proteins in different orders and make custom-built proteins for a particular situation. So if the cell has to adapt, or if you have different types of cell, then they may make two different proteins from the same gene. So saying that we got 22,000 genes doesn't mean we've only got 22,000 proteins. We actually make a few more than that. But proteins have become the sort of main structural component of cells. So obvious proteins, we have a keratin, for instance, in our hair, and collagen, the long fibers in our skin. And the collagen fibers are very useful. They're also both of those are in our bones as well. So we use those particular fibers as structural components. And then there's tubulins and actin inside your cell, which are kind of used as a skeleton for your cell. So actin plays a big part in our muscles, for example. When we contract our muscles, two of these actin filaments walk towards each other. Literally do this little thing with feet, and they walk towards each other. And pull the two ends of the actin filaments in, shortening the cells, and that's how muscles activate. That happens billions of times a second when you squeeze your muscle. I'll show you a little picture of that later on. This is the instruction set. So does anyone know an entire instruction set off by heart, like a Z80 instruction set or anything? I guess you guys are all too young for that. But you should probably know that 21000 means load HL with 0, for example, or 90 in x86 means no op. This is the instruction set of the cell in terms of making proteins, anyway, and and it's kind of divided up into the individual amino acids, but it also has things like stop. So we have an exit one in our code and various other things like that. So mostly this is a code that's been around for a very long time. So about sort of 3 billion odd years this code's been around. So about the first billion years of life, things were pretty hard for life. They had sort of great big meteorites landing on the Earth daily. The seas were boiling, and if you had any water at all, and life was pretty hard for life. But life evolved this fairly complicated mechanism about 3 billion years ago, and then it was so good it kind of lasted. It's been like Fortran. It was it works. Don't mess with it, whatever you do. So no, this stuff has been around right from the word go, and it hasn't really changed very much. So everything from the smallest bacteria to great big lumps of meat like us, we all use pretty much the same code, and that's how it's so easy for viruses to go between species, for example, because we share a lot of the mechanisms of living with so many critters. But all these amino acids do different stuff. So things like glycine is kind of a little short one. Arginine is like a positively charged double-headed finger. Lysine is a positively charged long finger that kind of reaches into holes and pulls things out from time to time. Then we've got sort of more structural things. Some of these are hydrophobic, so they tend to go into the inside of things. Some of them are hydrophilic, so they tend to go to the outside of the protein, interact with the water, and various bits like that. So they all kind of have their own things. So histidine is an interesting one, for instance, because it can't make up its mind whether it's positively charged or neutral, and depending on the pH of the water, for example, then it'll change. And there's various proteins which kind of, if the pH is such and such, they'll go and kind of open up and close down. So like the receptors in your tummy that give you heartburn, for example, those ones kind of use that particular aspect of histidine to kind of measure the pH of your pH of the thing of your tummy acid. So we can. There's lots of sort of mechanisms in there for doing different things. And out of this lot, we build a huge variety of different proteins. So probably the most abundant protein in the world is... Oh, yes, sorry.
How long have we known this?
So we've kind of known this, sort of, some of it since Victorian times. But the real details of it has been very recent. It's been, probably the last 20 years, just been completely rewritten what we understand about biology. So we've gone from sort of prodding rats with scalpels to sort of really starting to understand how life actually works in intimate detail. We kind of understand the small scale structure of things a lot better than we do the big scale structure still. So we kind of understand how. So for instance, my company works with. They've recently discovered how to drug a receptor in your lungs, which gives you cystic fibrosis, and they've got a drug which fixes the protein responsible for cystic fibrosis in your lungs, and they can cure about 80% of cases. And that's kind of, they've known about the protein for 15, 20 years or so, but they haven't found a way to fix it. And very recently they fixed it. That downside is they charge 200,000 quid a year to the recipient of the drug, and you have to keep taking it for the rest of your life. But probably that will change in the future. it's more of a problem with the patent system than with the drug, because it does cost a lot of money to make these things. But maybe we should be more have a different model for paying for our drugs. Anyway, so yeah, we can make a huge variety of different things out of this, including that little thing in it. So, for instance, I've got a dodgy channel in my kidneys, which means I get gout. So I don't excrete uric acid quite as well as everyone else in the room. So I tend to get gout. And we all kind of inherit different things. So we'll inherit different stuff from our parents, and I got color blindness from my grandfather, for example. That's a fairly straightforward X-linked thing, which means that only one of my color receptors in my eye is working properly. We get different variations in it. Of course, if we didn't get mutations, then we wouldn't get humans. We'd all be blobs of protoplasm right from the start. So thankfully, we get a few cosmic rays hitting the Earth and changing us in very slow ways. But of course, humans are amazingly similar. They're 0.00001% different, and actually humans are genetically almost totally identical. I think it's probably something like these guys are different. So arginine, lysine, histidine are positively charged things. And then there's negatively charged and hydrophobic and hydrophilic ones. So I think the asp is negatively charged. Those ones are probably negatively charged. So there's kind of three main things: charge, hydrophobic, hydrophilic, and they do different things. But different combinations of them make different shapes. So the two sort of big things we tend to use in proteins are alpha helices, which kind of just make little spirals, and beta sheets, which kind of zigzag backwards and forwards, making sort of sheets of protein. So, for instance, if you want to make a pore, then often there's kind of beta sheets which kind of go around the pore and make a big hole in the membrane. So there's different themes, and that stuff is conserved. So the first thing that it's like a design that lasts forever. So beta sheets came around, and then it was used everywhere, and so the genes kind of live for themselves in many ways. Oops. So you've probably seen these kind of videos. I'll keep the sound off because they've usually got an irritating soundtrack. Has everyone seen this video? And it's slightly exaggerated, but it's kind of fundamentally. So these are motor proteins in actions. This guy is kinesin. Kinesin's job is to navigate the motorways. These big things called microtubules are like the motorways of your cell, and they kind of act as channels that things get moved along in your cells. So this big ball that's carrying on behind it probably came from either outside the cell and it's carrying it in, or inside the place where all the ribosomes are, the rough ER and Golgi apparatus, and it kind of carries stuff between all the various compartments of your cell, and it does different things with it. The ball itself is made out of lipids, which I'll come to in a bit, and you see embedded in the surface of the lipids are various proteins which do various things, like they may act as an anchor point for this kinesin. When they reach the cell wall, those things will probably stick to the cell wall, and then there's various things like clathrin which can kind of push it out through the cell wall, and all these proteins have different things. So the microtubules themselves are made out of little proteins which, like Lego bricks, which kind of stick themselves together. You can actually see them grow under a microscope. If you've got a big, powerful microscope, you can see these things grow. And some cells actually move around. They go like this and walk, and they do that by the entire cell by growing out a little finger of actin that gets longer and pushes the cell out in front of it, and then another one goes like that, and the whole cell will kind of walk. So your skin cells are like that, your blastocytes and your skin cells. If you get a cut, they'll walk across the cut and heal the cut. Many of your cells in your body will move around on their own and are quite mobile. And there's lots of different behaviors in your cells, all sort of kind of made by a relatively small number of proteins. Oops, I think we're going to. Can we actually go on to the next slide? I think I need to click off that. Oh, there we go. Anyway, so this is a ribosome. This is the bit that kind of does the translation. So we have our 6-bit code, which represents a particular like PHEU, I can't remember which that one is, phenylene or whatever it is, and that's coming in there. It's going to kind of match up. So A goes to U, and U is the same as T in RNA. So U goes to A, C goes to G. It matches up, and then it kind of sticks the PHE on the end of the asp, and then spurts out a bit of protein, and it will kind of. So give an idea of the sort of time scale of this. It does about 10 bases of this per second per ribosome. You think, oh, that's really slow, until you realize there's about 1 trillion ribosomes per cell. And so this thing can really go fast. You can kind of, almost completely replace the entire contents of the cell in a relatively short period of time. So a fast-growing cell, for example, does nothing but this. It just makes new proteins for the cell in huge quantities and tries to create enough protein to then divide the cell, and those cells carry on dividing, for example. So bacteria, when they're growing, they're just huge factories for proteins. They produce tons and tons and tons of protein. And the proteins then go on to be structural bits, or they get to be chemical factories for making things, so there's a whole bunch of different things that the proteins do in your body. So I call the ribosome the high latency CPU because its tenth of a second is not brilliant. So 10 instructions per second essentially is a ribosome. Even though there's trillions of them, it's actually not that fast. There are much faster processes which happen in your cell. So for instance, your nerve cells are an example of that. When you prick your finger, for instance, then it transmits a pulse very quickly up your arm in a tenth of a second to your brain. And much faster processes happen when you do that kind of stuff. So the ribosome is the high latency CPU. It has feedback, so kind of satisfies at least one bit of the Turing complete thing, which is that the proteins themselves can then affect the production of other proteins. So we have these things called transcription factors, and they kind of come in great big blobs like this. Often with a bit of DNA kind of looping back to this site, and they may block or encourage the RNA polymerase, which is this thing from copying the RNA. So that's one way that genes are regulated. So an example is we'll generate collagen, for example. We'll keep generating collagen until we've got too much collagen, and then the switch turns off and it stops making so much collagen. That's kind of one example. But in practice, when most of the interesting stuff happens is when embryos are forming and when organisms are growing from a single cell. So you get your mummy gives you one cell. And then you divide and you divide and you divide and you divide, and every time it divides, those cells become slightly different, and those slight differences in the cells cause different things. So very early on, for instance, the cells making your hand, which starts off as a blob, starts separating, and your fingers separate out, and so that's caused by a little program of transcription factors which cause your fingers to form in a particular way. If you had a slightly different transcription factor, you'd end up with 6 fingers, not 5. It's just almost by chance that we have 5 fingers. It's just the way it happens. But there are some absolutely amazing things. For example, anyone notice anything about the length of my arms? They're the same. Amazing. So billions of cells have grown, divided to make my left arm, and billions of cells have grown to make my right arm, and the 2 arms have ended up exactly the same length. And we still don't really understand how that happens. That's an unknown thing, but the chances of that happening by chance is very small. And in practice, all the proportions of our body are all maintained. Have you ever seen the Michelangelo man with the circle round? That's everyone's like that. We all have a very fixed set of dimensions. We know that Henry VIII was 6 feet tall because a yard comes from the length of Henry VIII's arm. And so we know that size is the same as your height. And so that's all controlled by these programs in your body, and that kind of all happens as you develop from a fetus into a and keeps itself as you grow from a young man into a big fat man like myself. Then you maintain those proportions, and everything's controlled down to the last millimeter by your transcription factors. And that is a program. That really is a computer program, and it works very well. So your DNA controls those sort of low latency processes which happen. But it's not any good for doing like thinking, for example. Thinking is something organisms have only started doing fairly recently in the last few billion years, and arguably some people don't. Not going to talk about Brexit at all. So, low latency logic. So we need something a bit faster. We need to be able to think. We have ion channels. Ion channels are amazing things. So the ion channels are about as close to a transistor as you get in the world of life. What they basically are is you've got like a soap bubble, your membrane, and the ion channel kind of sits in the soap bubble, and it kind of lets things in and lets things out. And sometimes it just always lets things in. Sometimes it always lets things out. But the more interesting ones only let things in and out when certain conditions are met. So an example is your nerves. The way electrical impulses get transmitted down your. And remember that there's a nerve cell from the base of my brain all the way to the end of my fingers. That's how long those cells are. Then the signal gets transmitted along the cell by using these guys. So what happens is there's what's called a voltage-gated ion channel and sodium channel, I think, in the case of nerve cells. And when the voltage changes, it opens up and lets more sodium through, and the sodium changes the voltage. Sodium ions are positive; they make the thing more positive. It then causes the next one down the chain to open up. It lets in some more sodium, and so on. And whoosh, down the length of the cell, the signal gets transmitted. So it can, in a tenth of a second, it can open up all the ion channels from here to your finger. That's like 1 trillion ion channels getting opened up in a tenth of a second. So quite quick. So I think it'll order about sort of 100 million, I think, ions go through the channel a second. So it gives you an idea of how fast these things operate. And so not only do we have voltage-gated ones, which are essentially transistors, we have they act as amplifiers. So there's ones in our eyes, for example, when we see things, there's a big stack of little disks which are full of proteins, rhodopsin proteins. When one of the rhodopsins captures a photon, then it sends off a little signal which then gets amplified by another set of proteins, which gets amplified by another set of proteins, and that causes an impulse to go to your brain. It goes to the back of your head, where the visual cortex is, and lights up a whole bunch of neurons. Anyone do computer vision here? You probably know all this stuff. So we've been studying that process for quite a long time, and we kind of know how it all works. So these things open up and they close up, causing to signals. We could potentially use these things as transistors, for example, and they're very, very, very small. I'll come to biocomputing in a bit, which is kind of an interesting corollary of the whole thing. So for very fast logic ion channels. There's also a signaling mechanism which is also very fast called phosphorylation. And once you detect something on the surface of your cells, an example is a taste, or a smell. You smell rotting eggs, and there's a hydrogen sulfide detector in your nose. It opens up its ion channel when it detects when hydrogen sulfide comes along. You smell some hydrogen sulfide. It opens up. It generates a signal. That signal may, for instance, get either turn itself into a nerve impulse, but often it turns itself into a particular protein getting phosphorylated. It adds a little phosphate, group on the end of or many different places all over the protein, and then they act like AND gates and OR gates and things, and that signal gets propagated through the cell, just as a soup. It's computational soup, and it goes through the cell and does lots of different things in the cell. So they get a kind of cascade of things which happen. So do I have a picture of that? But yes, so we have various different things that can do, like phosphorylation, methylation. Methylation's very active in epigenetics, for example, and acetylation, all the other kind of things. But signals are mostly done with phosphorylation. So your immune system, for instance, having a response to a pathogen, for example, is mostly done through phosphorylation, and that creates a cascade of things which happen in your immune cells and cause you to get itchy nose or something if you've got an allergy. So these things have a sort of they're not sort of hypothetical things; they have a sort of direct effect. So we got CPUs, we got fast logic and networks for communicating in the forms of channels. How about the circuit board? So how do we build how does light build its circuits? Mostly it uses an insulating layer called the Lipid bilayer, which is these guys. So the most common ones you come across are soap bubbles. Soap bubbles are lipid bilayers, for example. It's not quite the same, but it's essentially the same thing, where some lipids are trapped between some water, for example, and these soap bubbles go around the outside of the cells, but also the internal compartments and liposomes and things within the cells are all made out of these little soap bubbles. And you never actually see it like that with nothing in it. In practice, these things are absolutely chock full of proteins. And the proteins have all kinds of different effects. So they can have like smell receptors. They can have bacteria, for instance, have chemical noses they can use to migrate towards particular food sources, or they're light sensitive. So our eyes kind of started off as kind of light sensitive bacteria, probably, and that kind of starts off as like bacteria having some advantage to steer towards the light, and ends up with fly eyes, for example, which are nearly as good as our eyes, but only in very, very, very simple logic to make a fly eye. Our eyes are a little more complicated, but not super more complicated than a fly eye. And we share a lot of the logic with flies that we have in our own bodies. So when we're studying humans, for example, we can't go and sort of do unusual experiments on humans. That's kind of not supposed to do that. So we don't say, please, sir, go into this chamber and we'll bombard you with ionizing radiation. So let's not do that. That's not considered ethical. But flies, they're fine. Oh, stuff the flies in here. We'll zap them with this, zap them with that. And so, we'll saw their heads off, or we'll cut all the legs off, or whatever. No one seems to care too much about fruit flies. So we learn an awful lot more about fruit flies than we do about humans, who we have to be a little more careful with, Depends if you're large social media companies, for example, who like to experiment with their humans. A/B testing. Don't take me any further than that. Anyway, lipid layers. So we have these like bubbles. They're full of proteins. The proteins communicate with each other. So now here we have a circuit board. It's got some components in it. Something happens on the outside. We kind of let go of some stuff on the inside, and the inside then goes all the way through the cell in a fraction of a second. So the whole state of the cell can change in a few milliseconds, depending on what's happened on the outside of the cell. So we can one of those things we can do with cells is we can kill them. And killing cells is an important thing, because otherwise we just turn into blobs of jelly quite rapidly. So actually, there's a thing called apoptosis, which is a programmed cell death. So we kill off cells when we no longer need them. Otherwise, we'll just kind of turn into blobs of cells, and that's kind of important. So when you get there's a gene called two genes called BRCA1 and BRCA2. The BRC means breast cancer, and they're DNA repair genes. When they stop fixing your DNA, then you get cancer. So there's a simple test that women can do to see if they've got BRCA1 or BRCA2, and if you have it, then there's various things you can do to stop you getting breast cancer. So Angelina Jolie, for example, knew she had BRCA2 and then had a mastectomy to stop herself from having probably about a 50-50 chance of getting breast cancer. That's kind of just drastic. There will be better things we can do in the future if you know what you have. I'll cover measuring your own genome if I get time towards the end. So power supply. So hey, power supply. So see, my son likes changing power supplies in computers, and so power supplies you get from your mum. Your mum gives you your power supplies, so you can blame mum for the power supply. The power supplies kind of started life as little bacteria that were living inside your cells, but they were useful bacteria. They're bacteria that gave you energy, and what they do is they manufacture the fuel for your cell. The fuel for your cell is a thing called ATP, adenosine triphosphate, which is a little, I think it's an amino acid with, well, no, it's a base, isn't it, with three phosphates on it, phosphate, phosphate, phosphate. If you chop off one of the phosphates, it releases a little bit of energy and makes everything a bit hot for a very short period of time. And the proteins have learnt to use that reaction to power themselves. So that little thing with the feet, what happens is every time it wants to move one of its feet, a little triphosphate comes along, goes bunk, and generates a little bit of energy, which lifts the other foot up and sticks it down in the other place. So that whole thing is powered by ATP. Does anyone like to know does anyone like to guess how much ATP a human makes during a day? It's the fuel that drives all your things. So. So you can I don't know. So, read the book. Anyway, the book says. I'm not that good a biologist. I'm a programmer, but. So kilograms, for example, or grams. How many grams of ATP do we make during the course of a day? 500. Any other guesses? No. So unless you know, of course, you actually make your entire body weight in ATP during the course of a day. Of course, you don't just turn into ATP. You recycle the ATP into so it's a kind of early renewable fuel. ATP gets recycled into ADP, which then gets put back again. So there's an interesting way that all happens actually, which involves an electric motor. So this is the electric motor. At least this is kind of two bits. So this is a internal combustion engine, and that's an electric motor. So what does that make that whole thing? It's a generator. But actually, the remarkable. So what actually can happen is ATP can come along here, it can spin this thing around, or spin the inside of it around, and it can generate electricity and charge up your mitochondria. But the normal way it works is that there's electricity inside your mitochondria, which are generated from breaking your Krebs cycle. If you know your biology, you're breaking down glucose into a bunch of protons, and those protons kind of accumulate in the membrane in the mitochondria, come out one by one through the little electric motor, driving the rotor around. And every time the rotor goes around, it grabs an ADP and stuffs a phosphate on the end of it. So it kind of recycles the ADP into ATP. And these things go trillions of times a second, and that's how come you can make God knows how many trillion. If you've got 30 trillion cells and you're making, like 1 million molecules a second from each of these things, then that's an awful lot of molecules of ATP. You'll make Avogadro number kind of 10 to the 6 times 10 to the 26-ish kind of numbers of molecules per second. You're kind of making with these things. And we've got a lot of these things. That's why we had to use little bacteria to. Plants have a similar one. So they have they have Ruby, I can't remember what it's called. It's Rub something or other, Rubisco. Rubisco in plants fixes carbon dioxide. So it gets the carbon from the atmosphere and turns it into carbon which the plant can use. So Rubisco in plants does a similar thing by grabbing carbon. All plants just grow from carbon. They grow from atmospheric carbon. So almost their entire contents comes from. And there's absolutely megatons of this stuff, of that particular protein in the world. And all the crops and everything we eat is made by Rubisco. And chlorophyll, which has another part. It's another part of the chain. So there's kind of a chain of different stuff which generates energy. Eventually, generates ATP for the plant, and the energy is used to construct new bits of the plant and stick things together. So this is the power supply. Oh, there we go. So follow some of these links if you can get hold of the. I won't bother to do that now. But, oh yes, we've got a little video of this thing in action. Again, I'm keeping the sound off. ATP synthase in action. So I could do the horrible American voice if you really want to. Oh, we do have the horrible American. There you go. So there it is spinning around. And this thing kind of changes shape. It kind of goes like this. And there's that little thing at the back which kind of holds everything in place while the whole thing spins around. And these little things are the ATPs and ADPs kind of coming in and going out again. And the coloured balls, the sort of ball pit is the lipid layer of your mitochondria. So electricity kind of comes out of, see the protons kind of coming out of there. They cause the thing to spin round, and it kind of crushes these things and stuffs the phosphate on the end, and does that a lot of times a second. Anyway, enough of that. So I can figure out how to cancel that. Anyway, so so that's kind of how life works. Anyway, so we got kind of an existing set of components. So interestingly, so how does this? Why are we having this talk in a computer conference? So we all like computers, don't we? Biology is boring. It's just sticking pins in rats and stuff. How does that apply to us? You know, how can we guys as programmers use this stuff? So and that's kind of the basis of all modern biology, essentially. So probably a big clue is just how many programmers work in biology, which is a very large number. So of new graduates, probably a substantial, I don't know an exact number, but a substantial portion of new graduates end up working in pharma industry, for example, or doing bioinformatics, which is the sort of the art of understanding biology through numbers. This is still a relatively new field. I found it completely devoid of technical skill when I first joined it. So we had sort of kind of ad hoc R and Python scripts which people ran on giant clusters because they were so slow. So it's very much an open field for good programmers. So if you're a good programmer, then you can eat these problems for breakfast. So you can turn something which takes a biologist a month to run into something which takes a few seconds or milliseconds even. There's still a whole ton of problems out there waiting to be solved in this domain. Can we actually use biology to make computers? So it's something I'm particularly interested in, although I can't physically do it because I don't have the skills. I want to know kind of, can we actually do something with biological components? We've got all these transistors, we've got all circuit boards, we've got DNA which can print as much as we like of. We've got robot labs we can use for making things. There's a whole bunch of tools we now have we can use to pretty much build anything we like in the biological space. And in fact, there are already quite a lot of hybrid silicon biological things out there which do various stuff, mostly in the sort of biological analysis space. I'll come to that in a little while. biological computing is kind of the field of using biological things to calculate things. So this is kind of a almost completely undiscovered field at the moment. It's kind of very, very, very early on in the sort of world of biocomputing. Is all quantum computing is getting all the press at the moment, and biocomputing is getting almost none of it. But in practice, biocomputing is a much more achievable technology than quantum computing in many ways. Anyone disagree with me? Anyone believe in quantum computing here? No one believes in it. There we go. So I'll do a comparison in a second between the two. Any questions in the meantime on what I've said so far? And no. Okay, I'll plow on. Something we've started to be doing. There's a bunch of guys in Oxford and Cambridge doing just this: is creating structures out of DNA. So DNA also folds in lots of interesting ways, and we can create bits of DNA because we can print DNA in limitless amounts. So the way we print DNA is we take a little bit of DNA, the bit of DNA we want to copy. We add a little what's called a primer, which is a little bit of DNA that complements the one tiny part of the DNA we want to copy. Then we add this like zip-like protein called RNA polymerase, and it kind of like Pac-Man. It kind of starts munching literally kind of munches the DNA, and what it does is it munches a bit of DNA and then squeezes a base onto the DNA and replicates the DNA. So it copies the DNA from one. DNA comes in a pair, so the first thing it does is it pulls apart the DNA helix. So it's like a little jaw thing, like a zip, that pulls apart the DNA helix, and then it adds two new bases to the inside of that, and then the two halves go away as two separate. So it kind of duplicates the DNA. It takes one half and copies that, takes the other half and copies that. It's actually a lot cleverer than that because it's got error correcting mechanisms. So there are error correcting codes embedded in your DNA. So methylation, for instance, where we change the DNA in very slight different bases of DNA in slightly different ways, tells us which one's the new one and which one's the old one. So the new one hasn't had a chance to be methylated. There's a repair mechanism that compares the shape of the two nucleotides that have been copied, and if they're wrong, if it's the wrong pair, it'll actually edit the DNA and snip out the wrong one and put the right one in. This is how clever proteins are. So there's that BRCA thing is part of that mechanism of repairing your DNA. So it literally fixes errors. But actually, the RNA polymerase, the copying protein is very good, and only makes about 1 error in a million. And then those 1 in a million errors are fixed by various parts of the error correcting pipeline. So we can print. There's a trick called PCR, and for PCR, what we do is we put this thing in, we put it in a tube in an oven, and we heat the oven up to almost boiling point. And when the water's almost boiling, the two halves of the DNA split apart. We can then latch onto this little protein will latch onto those things. When we cool it down, it will start copying those two halves. So now we have twice as much DNA. We cool it down, we heat it up again. Every time we cool it down and heat it up, we have twice as much DNA as when we started. So this is how we do things like taking the DNA out of Richard III and working out what his genome was, for example, or we take some DNA from a crime scene. We copy that DNA millions of times so we can analyze it. This has been so it won a Nobel Prize for someone sort of quite a few years ago, and it's the main analytical technique we have for DNA these days. So we can copy our DNA. It's like printing CDs. We can just make as much of it as we like. So if we can build machines out of DNA, we can just make literally as many of them as we like, almost for free, because they virtually copy themselves. And so we don't even need a chip factory to make these things. We just need an oven. And there's even some open source PCR machines which you can buy and put together with your 3D printed parts, and you can do it yourself at home and make your own DNA, copy as much DNA as you like. if you've got a good recipe for your DNA, you can just make tons of it. Literally, and so normally what they do, if you want to make literally make tons of it, they use big bioreactors. 300 liters, I think, is the standard one. So say you want to make a new set of antibodies for your body, for example, you stick them in a 300-liter bioreactor, go leave it for a few weeks, and open it up, and there's all your new proteins you want to make, or your all made by genetically engineered bacteria. So we can make biological bits. Washing powder, for instance, biological washing powder is just made by the ton, and that's made by bacteria typically. Beer, of course, is actually one of our older biological manufacturing processes. We use yeast to make beer, and more yeast, which is also quite useful. So we've been making things out of biological components for a very long period of time, but now we understand how they work. We can make very detailed mechanisms. So, for example, we can make particular proteins that bind to very specific parts of DNA. Those proteins can carry like a transistor or an AND gate or something, and then we can bind those AND gates or transistors to a particular piece of DNA. And they may be silicon ones as well. We could bind all kinds of things to the DNA, and once we've got our DNA template, we can then print as much as we like and automatically make circuits without having to put components into boards. So that kind of thing is probably going to be part of the future, whether it's biological components or otherwise, we'll see. So, and we have has anyone heard of Oxford Nanopore? We have a company in Oxford, and what they do is they use one of these pores, that makes a little hole in the membrane, and they feed DNA through it and measure the current as the DNA goes through the pore. And then you can read off the bases on the DNA. So they use it as a diagnostic tool to read your DNA directly from the current. So who can answer this question? I think beer wins. So we have. I think even if you didn't see the numbers, you probably think that beer wins. Socially, as a social lubricant, that's better than that. But in terms of thinking, then probably your beer can do more thinking than your smartphone, at least at the moment. And so even with 32 gigs of flash, which is quite an impressive technical feat. I just think how many transistors are in your SIM card. You can get sort of a terabyte hard drive, for instance. It has 8 trillion transistors in it, and but actually life has been doing that for a while, and beer even has been doing it for longer. So beer wins, obviously. So ROM-wise, we got quite a bit more ROM. RAM, we've got an awful lot more RAM. In fact, nearly as much. In many cases, more RAM than ROM. And the ribosomes are like, whether they're transistors or CPUs or whatever, it kind of beats 8 cores a little bit anyway. But admittedly, those cores are clocked quite fast. Question?
What if you're not allowed there?
There we go. Yes, that of course is a tragedy. But if you wait long enough, then beer will well, not hopefully dominate your life, but it'll at least be a very small part of it. So you can have too much of a good thing when it comes to beer. So I certainly don't recommend that particular course of action. Drink sensibly, as they say. Yes. Yes. So probably the beer because it's mostly the same. It's mostly the same DNA. So in practice, the beer probably has a lower entropy. Your smartphone can have different stuff. We could make beer which contains very high entropy. The problem with things like yeast is they fight each other, and it's a bit like corporations. One big corporation that makes a smartphone will end up winning, for example. And Yes, there we go. So the biggie tends to win, and yes, we need smarter beer. So that's the trick. Yeah, so it'd be nice if beer was had a bit more entropy. We could use it in different ways. Quite how you'd sort of connect your beer up to the outside world and ask it meaningful questions. You probably get the answer 42, of course, but you could answer different questions. So.
What will happen if in like 2030 Apple will release the iBeer?
The iBeer, yes. I'm hoping someone else will release the iBeer first. Maybe the Shandy Plus or something. There we go. So, any other questions? I do have a few more slides. Has anyone got the appetite for a few more slides, or shall I call it a day here? And okay, we'll do a few more comparisons. So the talk I did in the past kind of ended at this slide, which is probably the more interesting conclusion. But I'll kind of press on, and we'll do a few comparisons. Is anyone familiar with this book on the right? So if you've written compilers, you probably have to have read this book or designed CPUs. This is a book written by the guy who did the MIPS CPU, and the motive for making MIPS the CPU was to study the statistics of code and the computers. So one thing I did when I was at Sony making compilers was studying looking at running game programs, running MotorStorm, for example, over and over and over again, and looking at the statistics of MotorStorm and how many instructions ran at certain times, how we could, what was happening the most, what we could improve, whether we could change the compiler, which would then make MotorStorm run faster. And these kind of statistical approaches we used to kind of analyze CPUs. We didn't trust the manuals, so ignore the manuals at the bottom there. The guides you get from Intel and AMD are only approximate. In practice, if you want to find out how a CPU works, you have to write lots of random programs and run them and find out how things actually work. And so latencies of instructions and that kind of stuff. So you really need to know a CPU in incredible detail, probably more detail than the maker of the CPU chip. Was certainly one person working on CPUs are big projects. So when we were designing the cell broadband processor for the PlayStation 3, for example, then it was done in Austin, Texas, and in conjunction with IBM and Toshiba, I think. And it was like a sort of probably tens of thousands of people working on that chip, and one person had no idea how the whole thing worked. One of my friends, Masa, who was kind of in charge of part of the project, when it didn't come in on time, in Japanese culture you have to show remorse, and one of the things Japanese do is they shave their head. They used to do something a bit more drastic when you failed your lord, but what Masa did was he went out and shaved his head, and this is kind of a kind of way of showing that despite your best efforts, the project hasn't come in on time. So it was a very difficult project. So it kind of shows you how complicated CPUs are. And modern CPUs are rarely complicated. In fact, they're probably more complicated than cells. And life forms. Possibly not as complicated as, although the cells themselves are relatively simple, there's only 22,000 proteins, they self-organize themselves in lots of really complicated ways. And that's the thing we don't understand yet, is how whole big ecosystems of cells organize themselves into being fingers and noses and nervous systems and eyes and visual cortexes. Those kind of things we still don't understand, because actually there's very little complexity in the cells. Surprisingly little complexity, and how it is that this complex thing, which is a human being, gets made, is we don't understand. And it's pretty much the same with computer chips. We've made really complicated things. We've stuck them all together, and behold, they actually work. But it's kind of a surprise that something that complicated actually works. So in the biological world, we got my friend. Cell Biology by Numbers. This is a book which is written pretty much with the with the same kind of philosophy. Again, ignore the manuals. In the case of life, there are none. We're in the process of writing them anyways. Yeah, unfortunately, life didn't come with a manual. Blame God for that. There was no manual. So here you are, Eve. Here's the manual. Whoops, lost it. Bioinformatics requires intricately detailed knowledge of computer systems and running endless stats, and this is where the prodding the fruit flies comes into it. And what I do in my day job is I look at millions of humans and compare their genomes and see how various things about the humans changes with small changes in the genome. So we do what's called GWAS studies. So we take everyone in this room, everyone spits into a test tube. We read all the differences between all the people in the room, which is very cheap these days. And if this bunch of people get colds and this bunch of people don't get colds, then we can learn something about what gives you a cold from those data. Quantum computing. So I'm not a quantum computing believer. I must admit. My name's Andy. I don't believe in quantum computing, despite being a physicist. I'm sure you can do quantum computing, and we can do quantum computing at the moment. But actually making it useful is maybe a more of a challenge. I'd love it if it was possible to get something for nothing, which is essentially what you seem to be able to get with sort of every time you double the size of your quantum computer, you end up with universes full of extra computing power. That'd be great, and maybe we could do something interesting with that. But in practice, there seem to be some flies in that particular ointment, not fruit flies either. We shall see. It'll probably come in about the same time as fusion power, as we know. So about the same timeline as, or the James Webb Space Telescope. Another project that's taking a while, rather a while. So biocomputing, we have the bits. So the gods have provided us with most of the technology. We can use quite a lot of that, and we can print DNA as much as we like. We already have DNA printed on chips, which I'll show you a couple in the next slide. So pretty much we can kind of do it. So if we had a biocomputer, we could get 10 to Avogadro numbers of components into a PC case, and sort of build Google that requires the same power as a light bulb, rather than Google that requires 5% of the energy on the planet. These are hybrid silicon DNA chips. Very, very, very common. They make absolutely millions of these things. So my guess is that these are the things you spit on. So you spit into your tube, and they put the spit on one of these things with a few chemicals. They do the PCR trick where bits of the chip are primed with particular sequences that you're looking for. So this one's like the top left-hand corner is ACCGTA, and the bottom right-hand corner is ACCGTAGG or something. And each of those bits corresponds to a known part of the human genome, usually a part you actually care about. And these are called variants. What I work with day to day. And then what happens is you give it a bit of chemical treatment. It replicates the DNA with a fluorescent thing attached to it. You shine some ultraviolet light on it. You can then read out which one of those things is a yes and which one of those things is a no. Gives you a pattern of bright and dark dots, and that tells you what differences are between two genomes. That's probably a reminder that I should be finishing this lecture fairly soon. So is everyone still interested? Good. Hooray, here we go. So these are existing chips. If you go to, so you can actually use these yourselves. So these microarray chips are cheap as chips. They're about 20. If you happen to be 23andMe or Ancestry.com, you can probably buy them for about $1 each. they'll charge you about sort of 50 bucks, I think, to 50 euros. I'll stick to euros because it's a hard currency. And 50 euros, so 50 euros ago, you will get there. What they do is they'll tell you all kinds of stuff you may or may not believe about your genome, but also they'll give you the opportunity of downloading your variants. You can download a thing called a VCF file, just a good old ASCII TSV file, like almost all bioinformatics files are, and you can have a look at your genome and compare it with other people's genomes. There's even a website called. I'll come to that in a sec. But you can do that fairly easily. If you really made of money, if you're Bill Gates, for example, you can have your entire joint genome sequence. That costs you a whole 500 euros or so, depending on how much depth you sequence it to, and how much you want to believe the answer. So you can have your entire genome sequenced for about 500 bucks. So there's some projects like the 100,000 Genomes project in the UK, and a few others like UK Biobank, who are sequencing literally hundreds of thousands of people, and we're just starting to get that data, and so it'll tell us a lot more about the genome as we go. So we can do that, and there's big parts of your tax money are being spent on this at the very moment, and when it's done, we'll know an awful lot more about genomes. You can also nanopore. You can get a thing called a minion, which plugs into your USB port, and you put your DNA in that, and it reads out directly the DNA from your thing. You may or may not believe the result, but they've got some quite good software which do base calling on it and things and tell you the variants. And then PCR is the cheapest of all, so that's the heating and cooling thing I mentioned earlier. So with the right chemicals and heating and cooling, you can it doesn't give you like your whole genome. It gives you just like one or two spots in your genome, and you can amplify bits of your genome and have a look. So they do this for things like DNA fingerprinting and stuff like that. So it's a very cheap technique. They used it out in Africa for looking at the mutation of HIV virus or Ebola virus in the bush. So you can literally, sort of, no electricity. You can sit in a grass hut in Guinea-Bissau or something, and you can actually sort of tell quite a lot about the sequence of an HIV virus with a relatively simple machine, and it can cost you about 50 bucks to make. Actually, Africa's getting to be quite a good place for doing genetics, because a lot of places like Uganda and Rwanda have invested quite heavily in data centers and equipment to do sequencing. In many ways, Africans are a bit more interesting than us inbred Europeans, because a very small number of according to the Out of Africa theory, a very small number of Africans said, "Oh, we're off on holidays," and they ended up in Egypt growing wheat, and then populated almost all the rest of the world. So in case of Europeans, we're actually very inbred, and we only go back a few generations, whereas Africans go back like millions of years, and you can kind of find variants in Africans which are much older than variants in Europeans. So they have a much more diverse ancestry than Europeans do, who are all horribly inbred locals. Speaking for myself. Share your DNA. So once you've done your 23andMe trick, then go on to OpenSNP. There you can share your DNA with your results of your sequencing with other people, and you can kind of see other people's sequences. You can write some software to compare your sequence with someone else's sequences. There's lots and lots of papers and lots of programs out there you can use to process your DNA data and do your own GWAS studies and things. Often they get you to fill in a questionnaire, and the questionnaire answers essentially are things you can use to measure your, like the eye color, for instance, is something you could compare with your genome and find out what causes your eyes to be blue or green or purple or whatever. A bit of alien DNA, maybe. Strange shaped eyes. There you go. So, those kind of things are relatively simple and peripheral. but you can share your DNA and use that, do your own studies to find out more about your DNA and other people's DNA. Look at your family. Look at the similarity between members of your family, for example. Maybe be careful when you do that, because your great-great-granddad may not actually be your great-great-granddad, as case might be. You have an option of doing that kind of stuff. So it's an interesting project anyway. So if you're interested in genetics and these kind of things, there are a lot of software websites where you can share the software and you can share the data and learn more about it. Anyway, I'll shut up now. And so, any final questions before we wrap up? And one at the back. And
two questions. One is, if you share your DNA like that,
yes.
Could somebody then basically print that DNA and leave it at the crime scene?
They could do, yes, and but they'd probably have to be quite rich. So yeah, DNA evidence is not the ultimate truth. Particularly when generally law and smart people tend to avoid each other, as certain presidents of certain countries might be able to demonstrate, and so smart people can always find ways of getting around these things. But you have to be quite rich, and you'd have to do all that stuff undetected in a very cunning way. The complexity of doing it will probably give you away. So be very careful how you do that stuff if you're planning your perfect murder. Another one.
That's a completely different question. So how big would a biological transistor be compared to a silicon one?
So I think their order is about sort of 4 or 5 nanometers. So they're probably, you could definitely make a silicon transistor which is that big, and it would probably be faster, but it would probably use more energy, and building 3-dimensional structures of transistors is at the moment kind of just about doable with flash memory, but very hard to wire them up once you've made them. The nice thing about biological components is they do their own assembly, so you don't have to pay slave wages in South China to get people to stick with things on circuit boards. They'll all arrange themselves. So,
talking about how interesting the morphology is fortunately so well controlled.
yes.
I saw a program a long time ago about how gene activation is controlled by gradients of
Yes.
chemicals.
Yes.
So you can get these like two-headed bees or bees.
Yes.
It's still fascinating that the implied level of precision for measuring that gradient
Yes.
is incredibly fine.
Yes. So there's we know most of what we know about those gradients and things from fruit flies, because they're the most studied things. So there's a bunch of very early proteins which are involved with patterning like notch, for example. It's called notch because certain flies had little notches in their wings. That's how they discovered the gene, and so they wanted to know what notch did. Actually, notch patterns the segments in the fly, so the fly gets divided into segments when it starts. There's a really good book called Making of a Fly, quite an old book now, but it kind of goes through all the steps. And these days, with modern microscopy, you actually see them all happening. So with really good light microscopy, you actually see all the cells dividing and how they arrange themselves in the embryo. So you get sort of migrations of cells. So mussels are very interesting. For instance, they start off with a single cell that elongates itself and spans 2 points inside the fly, and then as the fly grows, it keeps elongating and recruits other cells around it to form the muscle, and that they'll form the fiber of the muscle and join two very specific points of the fly to each other. So we know a lot of this stuff about how things work, and particularly fly brains are an interesting one. Oh, fly brains! I love fly brains. There we go. There's a very interesting project recently to map a fruit fly brain and work out exactly what it does. There's about 5,000 neurons in a fly brain, so we can pretty much get a night. We build our own simulated front. So if anyone wants to help with that project, there's a big project to simulate a fly brain and make a working model of a fly brain. So you guys in this room are probably fairly well placed to get involved with that kind of volunteer science project. You never know, it may change your lives. So yes, all developmental. I find embryology, as gruesome as it sounds, to be a very interesting thing, and how our structure of our bodies gets formed is a very interesting process. Right, so are we done? And I can't. Oh, we've got some. Oh, yes, you're in the light.
Yeah, sorry. Isn't sort of rule one of the non-spherical planets of the grey goo, do not let your computer self-replicate?
Well, too late. Life has already turned the planet into grey goo. Actually. There's a whole ecosystem out there, and life has had a hell of a long time to get really good at doing it. And if we made our own life, just keeping it alive is a major challenge. Things like biocomputers we could make, but we have to keep real life well away from them because they just eat them, literally. Or you could eat your own computer. So we have to keep it well away from real life because real life is really aggressive and really adaptable, and things we make ourselves wouldn't be that adaptable and aggressive. And that's probably true of nanomachines as well, because sure as hell some form of life will find some way of eating them. Yes.
You said that the circuits may print themselves. How do you power them?
The ATP is the traditional way of. So mitochondria are kind of ready-made batteries for cellular life. You may need to feed them some precursor chemicals to get them moving. Our cells usually run on glucose, so we have a whole series of proteins which break down glucose and turn it into protons, and the protons then make the ATP. So you could just put a whole bunch of you can literally just put a whole bunch of chemicals in a bucket, and it will carry on doing that. But keeping the mitochondria alive is like with all life, is a bit of a challenge. But we may find some other way of doing it. But ATP and mitochondria are kind of an existing way of doing it, so we could power biocomputers that way. Any others? Yes.
Just asking, if you had a PC which was made on life, and you had a dog, after the dog,
Yes.
would it be like someone's just in the middle of like playing Sam Rattle or something?
And if
Half of the dog would eat my computer.
yeah, you need to make sure your dog doesn't eat your computer. That's a really important thing. But dogs are quite a bit bigger life forms than bacteria, but bacteria will probably get there first. And, but definitely watch out for the dog and with your hamburger computer. And
So could we imagine a biological computer virus?
oh yes, absolutely, yes. And it's like life may get there first and make one for us. So yeah, you could definitely. Of course, that virus is actually quite a good way of programming things, essentially. So, the EEPROM programmer of the future essentially is a virus. We can change our own DNA with CRISPR and things, and so viruses are a very good tool for doing that. They've evolved in various complicated ways to be able to change the DNA of a cell.
So you talk about Google in the bucket. So what's the cost per instruction? Do you estimate for biology?
Yes.
Say the gigantic cost of very low.
Yeah. So could be very low because in a conventional computer you've got millions of transistors all collaborating to make one instruction execution. That's kind of how come we can have 3 gigahertz clocks. So we got millions of transistors all have to go clunk. It's a bit better with dynamic logic, but with static logic, then we have D flip-flops which consume a huge amount, and DRAM, talking to your DRAM takes a lot of energy. So biological stuff, if it's talking literally just exchanging a few molecules, is like femtowatts or femtojoules. It's absolutely tiny amounts of energy potentially to make a switch. The ion channels, for instance, switch with almost no energy at all. And a lot of these things are self-powering. So the DNA replication is actually self-powering because the phosphate backbone of the DNA contains enough energy to drive the process. It's kind of one of the side effects that the DNA itself contains enough energy to replicate itself. And
did anybody manage to use the cell for mathematical computation?
So there's a guy in Bristol, curiously, out at UWE, who uses slime molds to do calculations. So that's kind of sort of the big scale, I suppose, of and of course the neural networks were real neurons, right? Back in the 50s, people were kind of sticking pins in real neurons and measuring currents. So people were sort of building networks of real neurons, but it's not particularly a good or efficient way of doing it. It's still probably it's not bad, but people have already started thinking about this thing, and so there's a guy at UWE who's doing slime mold computing, thinking about computing in non-traditional ways. We should all think about computing in non-traditional ways. There may be different ways of doing it. So anyone else hiding in the gloom? Right, I better let you all go. And thanks very much for coming. I hope you sort of go away and try some of this stuff for yourselves. Bioinformatics is a very interesting world. The company I'm working for is hiring, for example, Genomics PLC, and many other companies in Oxford as well. If you are in Oxford, come along to our biohackers meeting and see for yourself. Thank you.